This is PharmaForum web editor Nicole Raleigh, and I am at LSX World Congress in Lisbon, Portugal for the final day of the conference. And with me today is Roi Raz, who is CEO of ReBio Pharma, a preclinical stage biotechnology company that has developed SiSu, an injectable bioresorbable sustained release delivery platform for ophthalmic therapeutics. Pleasure to speak with you, Roi.
Likewise, thank you.
Now, ReBio is advancing a new generation of targeted therapies designed to reach the right site at the right dose for the right duration. And uveal melanoma is the most common eye cancer in adults. Still, no approved therapy exists for primary disease. So, Roi, I was wondering if you could tell us about the unmet needs in this disease and also the potential of ReBio's targeted therapy approach.
Sure, sounds good. So, as you mentioned, uveal melanoma is the most common cancer, ocular cancer in adults. It is affecting approximately 12 to 13,000 patients in the US, Europe, and Australia. I think the disease has two main stages. It's called primary disease when the cancer is localized in the eye, and metastatic when it starts to spread, typically to the liver. There are no approved therapies in primary disease, which is the largest clinical and commercial opportunity. The standard of care in primary disease management has been around for the last 5 decades or so, and it typically, for medium-sized tumor patients, are receiving a plaque brachytherapy, which is disc that is surgically inserted into the eye and is designed to radiate the tumor. And patients do reach decent tumor control with it, but almost all of them are losing the vision at the end of the procedure. And then the other option patients have, and that is for large tumors, so about a quarter of patients are just having their eye removed. So patients have basically no choice between losing their sight or losing their eye, which does not really affect their overall probability of developing metastatic disease. Uveal melanoma is one of the most homogeneous cancer that I'm aware of, at least. About 90% of tumors, carry GNAQ, GNA11 mutations, which can be targeted with PKC inhibitors. And so RBP01 is a localized and potent PKC inhibitor that is designed to treat primary uveal melanoma that is driven by GNAQ, GNA11 mutations, so more than 90% of patients. The purpose of RBP01 is to increase the exposure of PKC inhibition directly at the site of the tumor. So you can think about it as a targeted approach, right, at the site of the tumor and addressing the right molecular profile. There are several investigational PKC inhibitors that are administered orally for uveal melanoma. And I think the foundational work that some of our peers have done has really went a long way to validate the biological pathway, because we have seen kind of broad but modest tumor responses. So to us, that says that the tumors are definitely sensitive to PKC inhibition, but they don't reach insufficient concentration when you dose them orally, because they are protein-bound in plasma and you hit the therapeutic window, and which does end up with a lot of toxicity. So what we have shown in our development program for RBP01 is the ability of our PKC inhibitor to efficiently inhibit tumor growth in uveal melanoma disease models entirely. And so, but we saw that all PKC inhibitors reach this IC90, so inhibiting 9% of tumor growth at relatively high concentrations. And so we've shown in multiple animal models the ability of RBP01 to maintain very robust exposure so at the site of the tumor in ocular tissue that is orders of magnitude higher than IC90, and that is definitely higher than what you see with oral dosing. So we are really encouraged by this data. We presented it last week during ISUS, the International Society of Ocular Oncology, in a plenary session, and I'm super excited and humbled by the feedback that we have been receiving from the medical community. I think it speaks both to the unmet need in this indication, also to the quality of the data that we generated. And so we are going into clinical studies towards the end of Q2, so in the next few months. through an investigator-led study in primary disease, and we are really, excited about how this could be a transformative treatment for patients. We think that we can really reach a differentiated outcome.
It's just wonderful. Yes.
Thank you.
So, can you tell us more about the role of SiSu as a platform here?
Yeah, sure. So, going back 1 second, let's think about a few ways to get drugs into the eye. One way is taking eye drops, and that you need to take often and are working okay for diseases that are mediated in the front of the eye. But if you think about reaching posterior tissue, then you need, they reach in trace amounts. And then another route of administration is oral. But with oral dosing, you need to dose very high to reach sufficient concentration in ocular tissue. I think that idea is fairly well understood. We've seen, for example, we have several VEGF like orally available VEGF inhibitors, but we still treat wet AMD with intravitreal injections. So that is because typically when you dose orally, you hit the top of the therapeutic window, and you end up with toxicity. And so that was the case for VEGF inhibitors. That's definitely the case for PKC inhibitors. So SiSu is our in situ forming conjugate platform that is designed for programmable and precise drug exposure at the site of the disease or the tumor. We've been developing the technology since 2016, and since then we've created a library of drugs that address different tissue types, different dosing durations, different exposures that we want to reach. And what we've seen consistently in our development programs is the ability to dose within the therapeutic window as a monotherapy and even as a combination. We've seen very clean tolerability, to be honest, and this is not very surprising because this is the core IP that relates to the chemistry process that we do to design our drugs. And then the third element, which is incredibly important and even more so in oncology therapeutics, is the ability to dose and redose between 1 and 6 months. If you think about Ozurdex, which is the highest selling sustained release therapeutic for eye care, it is providing drug for 3 weeks but stays in the eye for 3 months. So if you think about having to redose, and that's how you do typically with cancer patients, you need to titrate up or down or take on and off therapy, then you would just end up with your vitreous clogged. And so we have shown the ability to safely and repeatedly dose with RBP01 and with other drug candidates. So super excited about it.
So you should be, yes. Okay, so on that excitement, why are you at LSX this year?
Yeah, so we've seen the vibe at LSX has been incredibly strong. We've been having really interesting conversations with colleagues, with peers, with investors, and we've seen, I think there is kind of an interesting move towards validated biological pathways. So kind of moving from the drug discovery, which makes sense in terms of the deal making that we've seen over the past few years. And so to validate biological pathway that can reach clinical validation relatively quickly. So I think in the context of ReBio, that puts us in an incredibly, that makes our conversations even more interesting because we feel that the biology is relatively well understood. We have a clear path to go into the clinics and to actually generate comprehensive characterization of P01 over the next 24 months.
Well, thank you for your time today, Roi. It's been a pleasure.
Thank you. Thank you.